Gomez-Lobo. Obviously, we're recording this webinar. I'm a pediatric gynecologist. I'm a member of the Pediatric and Adolescent Committee, as well as the Science Committee. Shuo and Ariella Shikanoff are the leaders of that committee. And what I wanted to tell the group was that in 2019 at the Oncofertility meeting, Francesca Duncan came up to me and said, you know, we've got this precious tissue from the National Physicians Collaborative, and we really should do something and figure out what we're going to do so that it gets the maximum use. And we came up with this idea of creating an image bank of tissue slides. And so they transferred all the H&E stain slides of all the tissues that they had at Northwestern to the NIH. We then added tissue that we collected at Children's National, and in Pittsburgh added tissue. We've had tissue from a few other sites. And so we've created this wonderful resource of images that are in a Halo cloud-based system with associated clinical data. And this is the page that announces this that I'm sharing right now. If you go to, if you Google me at NIH, you will find this page. I will put this in the chat so that people have it. And you can put in a one-page proposal and we will share images with you. And so that brings me to Sanju, who I'm going to stop sharing now, who approached me and said, you've got this resource. We really want to do some research and AI on ovarian tissue imaging, and we'd love to see some of the young tissue. So Sanju got his PhD at combined University of Maryland and NCI, and does research on ovarian aging and aging in general, and it's all sort of computational research, which I don't understand a lot being a clinician. And so when he told us he could do this analysis from just images, H&E images of the ovary, we were pretty excited, and we really thought this should be shared at large with other people who are involved in ovarian science. So with that, Sanju, I'm going to stop talking and I'll let you share what you were going to share.
Thank you so much, Veronica. I see that Francesca has joined. Is it the Francesca you were referring to? Veronica, you're muted.
Francesca, are you here? Hopefully it is her, because
I don't think that is Francesca Duncan, maybe another Francesca.
Okay, wonderful. Well, we'll make sure that she hears it.
Thank you so much, Veronica. And thank you, everyone, for really your time, and Veronica for this opportunity. Today, I will share something, a little bit of a journey for us, why we are here, and why this data set excites us, and what we are trying to do, and try to convince you that how we believe that histopathology images could really be quite exciting with the recent set of AI new tools that are coming out. We call this project in our lab The Shape of Aging, okay, inspired from the Ed Sheeran song. And today I will talk about overview of our methods, what the previous methods that I was developing, and almost like a journey of why from a non-believer of histopathology images that I have reached here, where primarily my lab focuses on histopathology images. One tool that we call EC-Path focuses on tumor to predict ecDNA, one that we have extended to normal tissues to predict telomere length, and then identifying critical age periods of each tissue, where ovary is our poster child. So we will really not focus on ovary till the very end almost. So please bear with me. I really want to share with you some of the technologies that we are developing, and would love to hear actually feedback, because we are sharing a large part of this presentation for the first time. So if something is unclear, it would be very helpful if you tell me which parts are unclear, and I can try to do better. Okay, just to give you an overall start of that, during my PhD, I actually did not focus at all on histopathology images. My focus was primarily on precision medicine using omics, where I developed multiple different publications. We had 20 different publications or so, but just noting a few, like Perception, this was the first single cell based patient selection method for targeted therapy. Like it was one of the NCI milestone of the last year. Deep Target to find targets for cancer drugs. how can we better use the tumor mutation burden as a biomarker for immunotherapy, and a few more. However, our journey of histopathology really started something like this in 2023, where I was done with my PhD and I still had some time remaining, okay, at the end of my PhD. And my boss like, okay, Sanju, you can do whatever the hell do you wish to do, okay, don't bother me, that's it. So I'm like, okay, what is the richest data that is there, most amount of data, how we could make sense out of it. So that was when I went to TCG and got all the histopathology images, and each histopathology image is like 2 GB. Okay, so you can imagine that each RNA-seq that you get from bulk RNA-seq is just like 20,000 vector, right? So it's actually just in KB size. On the other hand, each histopathology is like gigabyte size. And I tried to do is this, try to predict the bulk gene expression of the tumors directly from the histopathology images. And what we saw is something really interesting, that we can almost predict 3,000 genes bulk expression just from histopathology images with a correlation of 0.5. Okay, I will show you a little bit more data today because I want to convince you that this is real and this is not just me claiming this as like high computational stuff. Okay. But what this really started, that this is while it's published now last year, is that now my colleagues took this and tried to predict methylation data, that now this is the state of the art right now to predict brain subtyping. Okay, so this is predicted methylation and then tried to predict brain type subtyping. And now there is a whole field focused, not just my colleagues, now also a bunch of other individuals, bunch of other labs trying to predict spatial expression learning from histopathology images, where you have parallel spatial expression and H&E images. But what we are doing is following these 3 green boxes. I started my lab in 2024, and we developed this method that we call EC-Path, where we are trying to predict ecDNA. Okay, I will give you more depth of this. I just want to show you the breadth and overview of this. And this is right now in review in Nature, this paper in revision, but then we extended it to normal tissues and predicting telomere length, and now trying to understand how tissues are changing as they are aging. Okay, so this is the overview of it. So let me go depth of it to try to convince you that what I'm really saying is some of it makes sense. Okay, for a subset of individuals who don't know what is digital pathology, if there are some, I just want to take 30 seconds for this. It looks like this almost. Almost 100 years ago, we realized that we can put a piece of tissue on a glass slide and look it under the microscope with 2 stains that can basically stain the nucleus and the cytoplasm of the tissue. In next 50 years or so, we started scanning some of it, and we realized that we can create digital imaging of that. And then we started adding omics data to it, and we realized that, okay, you can have imaging as well as you can have the omics data, and it can help you try to make predictions and do better clinical service to the individuals. But what is really happening now, that these digital images were very rich, as I said, almost 2 GB each one of them. And what started happening is that in the last 2 years or so, there are multiple papers that came out that we call almost pathology foundation models that tried to take these images and learn quite a bit from this. So there are, you can see these are very high impact papers that came out. Meta released a few papers, Microsoft released these few papers, because simply our ability to learn what we can from these images really increased. Let me try to give you a little bit more what is really happening in a nutshell in these papers. But the first thing happened is this almost 5 years ago of deep learning breakthroughs, where we learned that there are methods, deep learning methods, that can learn quite a bit from these images. Then there are larger data sets that started being available of pathology images. Then there are multimodal data, not just pathology images, but also a few other layers. And finally, the computational power started increasing as well, so that we can take these images and do the computation that was needed. So due to these 4 key breakthroughs, we were able to do quite a bit from these pathology images that I will show you in the next few minutes. Okay, so let me go a little bit deep. The first tool that we developed that we call EC-Path. So ecDNA is like in this electron microscopy images, you can see that these are your chromosomes, and these are the circular DNA that are there. Usually there are in 15% of the tumors that one see. These are more in pediatric tumors rather than in adult tumors. And this is an emerging anti-cancer target as well as biomarker that is emerging. There is right now no approved drug that can target the circular DNA, but there are companies that are trying to actually develop multiple drugs that can target this circular DNAs. However, it's very hard and costly to detect these circular DNAs because you cannot do it using just your regular exome-seq data or targeted-seq data. You either need whole genome-seq data as well as sophisticated methods to try to detect this extrachromosomal DNA. So our first goal was can we develop a method that can detect this extrachromosomal DNA in an accessible fashion just from histopathology images. So the first thing that we did is that we are not the first ones who are doing the histopathology or digital histopathology. So what we did is that we just picked up the standard deep learning methods that were there, and we tried to predict that can we predict ecDNA from histopathology. And in a nutshell, just to explain this figure, it did not work. Okay, so you can see that what AUC means here is that the maximum 1 AUC means that we can perfectly find individuals who have ecDNA versus the ones who don't have. 0.5 means it's just random. So you can almost see that most of our, in most cancer types in TCGA, our method actually do not work. Okay. So what did we do? We argued that the histopathology images are really has a lot of information. And if we want to pick up something so subtle as ecDNA, we would like to go and teach the methods to learn step by step. In computer vision, such methods are the following. For example, you can have same set of elements in an image, like for example, a bike, a road, an individual, and grass, but in different orders, and it could be a very different image, right? Like rather than pushing the bike, you are falling off the bike, even with the same set of elements. So the key idea is that you don't try to teach the model what is really happening, but instead you try to teach the model what are the different elements of an image, and you try to teach that specifically. And if you give a guided learning like that, you can potentially do a little bit better than just trying to teach the final outcome. This is actually not just in computer vision, also in question answering as well, models not trying to learn answer, but instead learning what is the reasoning steps, that how do you reach an answer. Similarly in video recognition as well. So what we did is the following. We argued that from a histopathology image, rather than predicting extrachromosomal DNA, we first want to predict the transcriptome, the bulk transcriptome that I just talked about, okay? And from there, we would try to predict extrachromosomal DNA. So as a two-step process. So I just want to show you that there are a few other papers as well that try to predict expression-guided prediction. We are not the first ones. And this is the overline that it looks like. You have a TCGA, we had 6,000 samples. We extracted, converted each image into a numeric vector. From a vector, we tried to convert it to a gene expression. And from gene expression, we would try to predict the ecDNA. So how does it look like? Let me skip this slide and show you this slide actually. Okay, so this is how the expression prediction look like, just in a high level, that in LGG or here in KICH, we can almost predict 3,800 genes with a correlation of 0.5, okay? But these numbers look very high, right? Initially, when you look at it. But when you realistically look at it, this is how the numbers look like. So let me give you one row to explain that. So in LGG, we had 500 samples in total. We tried to predict 17,000 genes. And if you take a 0.4 correlation, you can predict almost 12,000 genes, okay? Which is crazy high number looking. But actually, when you take a threshold of 0.75, the numbers look way more reasonable. It's like 26 genes, suggesting that we can actually not predict that many genes. If you actually take a high enough threshold, we are pretty reasonable. Like we can almost predict like 50 or so genes from histopathology almost perfectly. The key is that if you take a reasonable, a reasonable threshold, which we argue might be happening because maybe we are just learning the subtype of the tumor type that is there, right? And that's why you are able to put these numbers look quite high to you. So somewhere in between is a threshold that we may want to build on and try to utilize. So please remember that the predictions that we are doing is noisy. And that's why it's important that how we build these models are not just built on noise, but instead built on something more valuable and biology there. So this slide is something that, in my opinion, really reflects that how the realistic numbers look like if you try to predict gene expression. Now, with that in mind, now that we are predicting transcriptome with some level of imperfection, we argued that can you even predict ecDNA from the transcriptome? So we took true transcriptome, the measured transcriptome, and tried to predict ecDNA directly from there. So we built a machine learning model to do that in like 9 different cancer types where we had ecDNA label. And we can show that this could be done for like couple of cancer types, just in LGG, in GBM, and STAD, cancer types where ecDNA labels were more available. Not too much in cancer types where ecDNA labels were not available, as well as the biology of ecDNA is way more available and more argued to be more valuable in LGG, GBM, as well as stomach cancer. So we focused on these 3 cancer types to see that can our predicted expression also identify these ecDNA labels in these 3 cancer types. And indeed, not as good as the true expression, but with still a valuable stratification, we were able to identify the ecDNA from our predicted expression that comes from histopathology. So these are the ability of our model to identify extrachromosomal DNA directly from histopathology images. Okay, but what if our models are just learning the footprints of the gene amplification and not really the extrachromosomal DNA? So we also stratified our models where what if the model is just amplification of EGFR and cases where EGFR is not amplified, and we can show that we can stratify the ecDNA versus non-ecDNA even if the genes are not really amplified. So this is just more details of it. The paper is available, so you guys can see a lot of details, a lot of positive and negative controls that we put together. This is a slide more for computer scientists that we tried different methods, not just our method, and we show that our method right now across multiple different 3 key cancer types here is almost the best compared to if you go across multiple cancer types. But more importantly, I care about this slide, that what are the genes that were really used to identify the extrachromosomal DNA. And while our usual suspects, of course, come out of cell cycle stuff, but more importantly, I think the ones that are important, in my opinion, are chromosomal maintenance genes, DNA repair genes that are also supposedly are the genes that were used by our model to identify whether the tumor really had ecDNA or not in these 3 cancer types. Okay. This is where we really took a step further and asked that, okay, now we want to ask, how did morphologically our model identified whether the tumor had ecDNA or not? Okay, from pathology slides that looks like this. And we found that we gave our models and the high attention regions of our model to a pathologist and asked that, okay, these are the regions of the slide that our model is suggesting based on which the tumor has ecDNA. Can you tell us what is really happening in these particular regions of the slides? And she confirmed that these are the regions that have higher density of nucleus, looking undifferentiated, has pleomorphic nucleuses and larger nucleuses, suggesting that that tumors that have ecDNA are looking more undifferentiated and have a higher density of the nucleus potentially. And that is what the model is learning to identify whether a tumor has ecDNA or not. Okay. We tested it in an independent cohort of CBTN as well, where there were 1,000 individuals. And again, we're able to stratify the patients reasonable enough accuracy. Finally, we identify individuals that have no ecDNA label right now. is available, stratify individuals for survival as well, because in some cancer types, ecDNA can help you identify better prognostic individuals. Right now, we are working with 4 different places, one at NCI and 2 different companies that are working with ecDNA to target them so that we can help them identify individuals that have ecDNA. So this is where we are right now with this particular tool. That we believe that this could act almost as a pre-screening tool or a tool that can detect ecDNA from routinely available H&E images. It's a 3-step pipeline. First, it derives the morphological feature, then it predicts the transcriptional profile, and it uses that to predict ecDNA status. Finally, we believe that we have only reached a couple of cancer types where we could do reasonable performance. We are collecting even more data in other cancer types so that we can expand our method to other cancer types as well. And finally, also expanding it to predict the oncogenes that are present specifically in the ecDNA, not just identifying whether the ecDNA is there or not. This is the work that we are doing till now in cancer and tumor specifically. Okay, I'm going to take just a brief pause and actually ask if there are any questions or things that I could clarify before I go on to talk about more normal tissues.
Hey, Sanju, that is so cool. So I have a quick question for the transcriptomic data and your histology imaging. Do they have to be from the same level serial section, or same patient or same tumor, or as long as they share some common transcriptomic change, that is fine?
So, Xiao, we actually, some patients, we have multiple images, but we only have one biopsy. We believe it just has to be from the same biopsy. Okay, the tumor that has been taken, if it's different sites, then we cannot really map yet because we have not really tried to. We tried our best to keep it as matched as possible. Okay, so this is what we tried. If they are from the different tumors from the same patient, we are not sure that whether we would be able to do it or not, because we do see that during metastasis, some of the ecDNA is being lost sometimes. So we now have serial sections from small cell lung cancer where sometimes it's being lost, and we are trying to see if our method can really capture within the same patient or not. So let's see whether that works or not. At this stage,
Yeah,
it's all.
sure, gotcha. That makes sense. So my second quick question is, so when you mentioned the transcriptomic data, are they single, like 10x genomics data, single nuclei, or you need spatial transcriptomics?
Of course. Sure. We need bulk transcriptomics data that is very standard available in TCGA. Okay.
Gotcha. That's become bulk for now.
Yeah, we don't need single nucleus. We don't need spatial. We don't think there is enough spatial data for us to train such models. The way we are thinking about this is that we first train our model on bulk, then we go to spatial. There are some friends of mine who are building towards that. So that is a different direction to go towards. We can chat about it more. Okay.
Got you. Thank you so much. It's so cool.
Thank you. Are there other questions, other clarifications that I could give?
I have one question. So
Yes,
the transcriptomic data has to be from the same, what you call the patient, or it can be, you can predict the, the ecDNA from a different tumor, and the transcriptomic data is from different patient.
We actually don't need transcriptomic data, Raghu, from a new individual. We just need histopathology. Just during training, we do a transcriptomic data. Okay, it's the key for us that we just use it during training. Okay, we don't need it for the new patient. It's we train our model in a sophisticated.
Not for the new patient. Like for the training. I was asking, like, is the data coming from like from the same sample, or is it from just like a random tumor which is already in the database?
No. It comes from the same patient. It comes from the same histology, and expression comes from the same patient.
Okay.
That's the key idea. The key idea is that if you use just histology, it would not work. But we trained it to only focus on the parts of the images that are important to predict the expression, and then trained it to predict ecDNA. Okay.
Got it. Okay. Thank you.
No worries. Okay. So let's expand it and go take the next step to take it to normal samples. Okay. So we will now use a different data set that we call that is GTEx. Okay, GTEx is a project that was established in 2012 that is called the Genotype Tissue Expression Dataset, where the goal was to really, it was here actually, to understand how does adult tissue expression look like and what are the regulatory elements that are there. And the project started with almost 50 different organs that were collected from individuals who died in car accidents or in natural deaths. So these tissues that were collected were supposedly non-disease. I would rather say here that they are non-cancer. That's all one can say, because a lot of them have diabetes or different kind of diseases, and some of the tissues we can actually capture these different diseases and see that patterns as well completely standing. But this project has now expanded to also take data from children who unfortunately died due to different causes, as well as non-primates as well. So this is really expanding. So, but for today, I will only focus on the adult data. So it would be the following. We would focus on 18 tissues, okay? On an average, we have somewhere around, you can imagine, 300 to 400 samples per tissue. And we have their H&E images, okay, of these quote-unquote normal tissues of different ages. These individuals age from somewhere around 20 to age of 79 to 80, okay? And we have 1,000 individuals in total. What we also have from the same individuals is the following. We have their telomere length from the tissues. And you can see that if you put the telomere length on the y-axis and for each tissue on the x-axis, you can see that in some tissues, the telomere length is really decreasing very sharply, okay? For example, here in thyroid, telomere length really decreasing, but in other tissues, it's decreasing with a almost like moderate quantity low down. The key question that we asked here is that can we actually predict the telomere length from the H&E images, right? And you may ask me, why do I even care about this? Why is this even an interesting question? And this was just out of curiosity, that can we connect the structure of the tissue to the more like aging molecular factors? That was the key idea, okay? And we end up discovering a few different things, just stumbled upon them, okay? Let me show you what. So, okay, sorry to show this, but first, this is how the data, our pipeline looks like. We take a piece of tissue, we divide them into small patches, we convert them into numbers. So each tissue is converted into almost like 1,000 size vector, okay? So what first happens is the following. What we did is that we take this 25,000 tissues and put them on just a UMAP, okay? And you can see here that different tissues are just clustered separately. This is very intuitive because different tissues look differently, right? So if they're clustered separately, that makes sense from their images, that even a pathologist can say that, okay, this looks very different. This is separate here. It's fine. But what is really interesting is the following, in my opinion. When we actually took just one tissue, just like ovary, right? And we just threw it on a PCA, all the data. So each point here is the ovary section, H&E section, okay? What you can see is that the age is almost naturally clustered. That means that the individuals who are old are very different than the individuals who are young, okay? So the ovaries that are from old individuals look very different than ovaries that are young individuals. Again, actually, this is very intuitive in my opinion. Again, this is actually the fact that this looks very different compared to any other aspect that you can overlay on top of this. Like that what is the, for example, the ethnicity of the individual, how the tissue was collected, but the age is the one that really separates these individuals the most. This is the case for ovary and also many other tissues. We actually saw that this is the case for not just ovary, also for stomach, for esophagus, mucosa, colon, and not only that, many other tissues that here, except brain, many other tissues, age just naturally clusters out very separately for most of the tissues, which again, potentially could be intuitive because H&E of individuals who are old look very different than the individuals who are young. We argue that this could be a really interesting observation that could help us understand how the tissues are aging. And I will show you data of that in the next few slides. Can I ask a quick question? How much time do I have, Veronica?
You have till 5 o'clock. but it's flexible. So
Okay. What time is it right now, by the way?
it's 4:30. I think we're fine.
Okay, that's 30. So what we did is that while this is an interesting observation, and I would go a little bit more deep in that, is that we tried to predict the telomere length information for tissues. For example, in pancreas, we built a simple model that takes H&E as an input and build the telomere length as an output. And you can see that the true telomere is on the x-axis and the predicted telomere is on the y-axis. We can build pancreatic bulk telomere length model with the correlation of almost 0.5, in kidney as well, in prostate as well. So on an average, we can almost predict telomere length in the tissues where we had enough amount of data with a correlation of 0.5. Okay. And in those tissues, chronological age predicted it with a correlation of 0.2. So we can almost do 2.5 times better than what chronological age was doing. You can see here, the purple is basically where we can do way better than chronological age. While this is interesting, the key thing is for us is that we wanted to build this model because we want to identify cases where there is young tissue where telomere length is really very low, or we wanted to find tissues where are very old, where telomere length is really, really high. It could either be due to maybe tissues are damaged just due to autolysis. For example, here, there are cases of autolysis, right, happening in the tissue, or potentially due to different factors that are there. We believe that there's a technical noise that is in our data, as well as the fact that potentially individuals who are very good tissues are preserved, as well as they are going through healthy aging that we want to capture in this regard. Okay, so more important than actually just our model working is how our models are working. So we ask that, can we find what are the features that are really important for this model? And we try to interpret these features using different methods. So here are the results. First, we just ask simple question that, okay, tell me very simple properties like cell density, ratio of nucleus to cell. Are they different? Are these the properties that really were used by the model? And we saw that one key property that was really used by the model is nucleus to cell ratio. Okay, that if these are really high, the telomeres were really shorter. If you pick this up, you may know that this is actually a very important feature for senescence. Okay, when cells are going through senescence, their nucleus really becomes bigger compared to their cell overall cytoplasm. And this is one of the factors we believe that helped our model to identify whether there is a shorter telomere or a longer telomere. Again, we next ask, can you tell us like a pathology, like terms, like features that were really higher as well in the tissues where there were a shorter telomere? And we actually saw that our model is picking up just necrosis, damage of the tissue as an important factor that was telling us whether the telomere is higher or telomere is lower. Okay. We believe that there is a chance that we are not just picking up telomere, but potentially just also other molecular aging hallmarks that might be lower or higher, but we need to distinguish that. We are not at that resolution right now. We need other layers of molecular data to try to distinguish that. Okay, to summarize the telomere part, we believe that this is an extension of digital pathology to learn the aging process. Again, these methods are publicly available. You can look at this. This preprint is out as well. And this is a method that can help you determine the bulk telomere length of normal tissues from H&E slide scans. And the key idea is that it uses senescence-like features to determine the telomere length. Again, I will take a pause just to see that if this extension was clear or not, if this part was clear. Are there any questions? Okay, if not, I would actually jump on to the next part, which is how we can use it to understand, are there critical aging periods of tissues, and ovary being our poster child. Okay, so I have given a lot of, I'm presenting on ovary and oncofertility consortiums, of course. Ovary is certainly a focus of ours that we want to talk about and use the data that Veronica referred to. We realized and just stumbled upon this observation that As I showed you this data that ovary histopathology can very naturally separate out young versus old ovaries, right? We asked that if histopathology can do it, can other omics do it? And we saw that actually other omics cannot do this. So if we use this same exact individuals, 200 individuals and their ovary samples, and we have their gene expression, and we put it on a PCA, it actually cannot separate the young versus old individuals that well. We actually have also seen it from methylation. I haven't added that plot yet, showing that histopathology can capture, because potentially it's an accumulation of a lot of damage that is occurring during aging. But expression or methylation cannot do that. We are yet not sure why that is the case. One can argue that maybe gene expression is really a reflection of what has recently happened, or a bit more, but we think that histopathology can capture almost naturally young versus old individuals. But what is more interesting than even this is the following. We asked the following question. We said that, okay, this is good, but what if we capture how much changes that is occurring in the tissue on the y-axis, and the x-axis is just an age, okay? And we see this almost a bimodal curve, showing that the ovary is going through this major transformation. A lot of changes are occurring at the age of here, at the age of almost 35, 30 to 35, and again at the age of almost 55. Suggesting that morphologically speaking, there is a lot of changes occurring in ovary at this point and at this point compared to this point or here. And these 2 points align with our understanding that there is a major fertility decrease that occurs in this period, as well as a menopause occurring in the second period, okay? And we checked that, okay, if histopathology can capture this, can other layers of data can capture it or not? And we saw expression cannot capture it. We did the exact same analysis with the expression, and expression cannot capture this 2 peak that is naturally known previously. This suggests that histopathology can help us identify these natural changes that occurs in tissues as they are aging versus other layers of data. And we believe that this could be almost histopathology can act almost like an anchor for us to learn what are the big changes that is occurring in tissue in a 3D level versus the other layers of omic data, which can actually act as a complementary basis, and I would show you how. So what we asked next, that can we learn, now that we know this bimodal peak, what are the big molecular changes that occurs in these peaks? And here are the key idea that we ask, what are the changes that occurs here in this peak 1 versus trough 1, and find the genes that are specifically upregulated or downregulated using expression data that we had paired expression again from the same individuals. And we found that type 1 interferon signaling, cellular signaling to type 1 interferon, and a few other pathways that we are still dissecting. These is the inflammatory signals are the ones that are really high in the age of 30 to 35 versus at the age of 45, or we would say 40 to 45. We are yet really dissecting it. We have a lot of such data, actually. I'm just showing you one such plot, okay? We have compared these 2 peaks with each other, which is menopause versus fertility decrease, menopause versus before menopause, and just focusing on ovary. And we are just dissecting right now what are these major pathways, why that might be really changing. And this is completely unbiasedly. We never told the model that what is occurring at specific peaks, okay? It is really just identifying unsupervised. We believe that this is a method that can help us not just in ovaries, but also in other tissues, for example, to learn these major changes. For example, ovary has this bimodal curve, right? But when we actually went to other tissues, we saw different kind of curves. For example, in skin, we actually saw that there are apparently something we call almost like late agers, that a lot of changes occurs to them after 40, where maybe the collagen is being lost. So that's another kind of tissue-specific aging. Then we saw another kind of aging where there are a lot of changes that occurs in early, which is lung and heart tissues. Then we saw actually that there are tissues where there is changes occurring all over the place from age of 30 to almost 60. And finally, we saw some where actually there are a lot of changes occurring not at age 30 to 35, but rather from 40 and keeps going on further. We are still dissecting this, that how should we really interpret these tissues and tissue-specific aging. But all of this comes from just histology data and looking at that. What I want to convey here for this group is that we believe that histopathology data is really rich. Almost each image is like 2 gigabytes and has not really been fully utilized in understanding aging. There is a lot of confounding factors here, we believe, because these are postmortem tissues. There is autolysis that we need to capture and separate. But this is a really large scale data that could be helpful if it could be integrated with other data sets. Like I just gave one example. And if this could be done well, this could help connect the tissue structure dimension to the molecular property dimension, which we think a current piece that is missing in the aging field. This is the key idea that we really wanted to pass with the final piece of this presentation. With that, I want to give acknowledgements to my previous lab here in East Coast, actually at NIH, Dr. Ethan Rupin. And I realize how much green East Coast is now that I'm looking at this, that it was really, it was a wonderful time while I was at NIH, and I have a very, very high appreciation for NIH. I know we live in different times now, but I genuinely believe the reason for my scientific career being here, NIH deserves the credit for that, and Ethan and the lab where I had a wonderful time. And this is my current team here in the right. We are finding good Indian places here. So just telling you, if you are in San Diego, very welcome to host you here, as well as good Indian places that I can take you to, okay? Of course, my mentoring committee here, as well as our funding sources that has been listed. With that, thank you so much. I would stop and take questions, discussions, and hear what I could explain better.
Thank you so much, Sanju. My lab had heard this before when he told us why he wanted to use our images, and I think it's fascinating, and the opportunities are endless. So this is really exciting. Anybody have any other questions? I saw questions from Dr. Brown about who's an AI question. Can feature extraction and generative AI give pathologists a clue on determining the age from H&E images? And I guess you answered that question just now.
Dr. Brown?
Did he leave?
He may have left.
Yeah, okay.
I thought that it was a little bit early. I missed this question.
But I think you answered the question later on.
I see.
Oh, microphone's not working.
Yeah, Dr. Brown, I can actually answer that question in 2 phases. I'm gonna tell you, we did this as the first day we had that Chinese slide. Okay, we did this. I can send you the slideshow that we can predict the ages. It's not a problem, but we actually don't think that predicting the age, unless it's helpful to you, we are happy to do it. We will give you the model that can help you predict the individual age, but we don't think it's that helpful for us, for our science specifically. We kind of want to avoid trying to predict the age and rather try to capture what are the big changes that occurs in tissues. So we believe that aging is not really this number, right? Just 1 to 70 or 1 to 80 linearly, but rather we can almost feel it. Oh, the age of 31 is really important. Age of 30 is really important for my muscles, for my bone, for example, right? That I really want to build the muscle mass at this particular age. And so many different things that we know already that aging is so non-linear. But this has not been captured till now because people try to predict just the age of the individual. And we really want to avoid that question and do something complementary. That's what we were doing here.
Hey, Sanju, sorry, I cannot turn on my video because it's disabled by the host. So it's very cool, and I know Veronica has a lot of ovary histology images from different ages. I just wonder whether you can also consider to do some ovarian cancer prediction because you already have the tissue there, just in case they have some epithelium or other like early marker, even though ovarian cancer has been originated from the fallopian tube, but once they migrated to the ovarian surface epithelium, that would be so cool too, and I can see a huge potential application too.
Sure. Thank you so much for that note. very happy, guys, to work together. I told Veronica already that we are very happy to work together with this data. If there are ways that we could learn this prediction of ovarian cancer, it would be cool. I thank you for noting the challenge as well, that it's really the origin of it is from fallopian tube, as you're noting, but we're happy to learn, try to do it. We do think that this data could really almost act as a natural extension of our current works as well.
I have one question actually regarding predicting aging. Can we predict like during the aging process in the ovary, we sometimes see immune cell infiltration where you see phagocytic macrophages getting infiltrated near the damaged tissue or follicles. Can we predict a specific immune cell types like macrophages or phagocytic macrophages using this training model?
Raghu, I think I remember we briefly discussed this before, but the answer is that to be really sharp, I don't know yet, right? If that is really looking different. If they look different on the slide, then the answer is yes. If you give a paired set of immunohistochemistry and H&E, we can learn.
Okay. Yeah,
and then we can just go to these slides and just make the prediction and just predict how does these macrophages are really invading as the tissues are aging, right? And in cases where there is a pathology, how it is invading versus the ones where it's not. Where is it localized spatially? Are they localized around corpora luteum, where it's not cleared out very well, and stuff like that? So yeah, I'm sure that if the paired IHCs as well as H&Es are available, this could be learned or tried to learn at least. Yeah.
Yeah, we have actually, like I have recently done IHC for for macrophage markers actually, CD80, CD163, CD68. So we could able to see a nice infiltration of macrophages in the human ovary sections. So that can be used to train the model.
Yes, but do you need paired H&E data?
Paired H&E data. Yeah, I mean, it has only hematoxylin counter stain, and you have the the DAB staining, which actually stains the the macrophage.
Yeah, in that case, if you have the H&E plus this, then yeah,
both together. Okay, yeah. So then I have to stay in the side, the adjacent section with H&E staining also. Okay, yeah, we can do that one actually. Yeah.
Any other questions?
Also, clarification, guys, if something is unclear, you can really ask. We are presenting like some part, the shape of aging a little bit repetitive and parts because I'm presenting it for the first time. Okay. So I'm very happy for feedback if something I can try to explain better.
You know about this meeting, the Society of Reproductive Aging that's happening in San Francisco and San Jose in May? You may want to look at it.
Okay.
Because I think it's a new society. They're just getting started. I think Francesca's leading that. But I think this is really exciting. I mean, I came to the NIH in 2019, and at that point, people were starting to do single nucleus, and we were so excited about single nucleus. And then, we bought all these kits, and then all of a sudden you have to do Visium, and now you can do it from images. I mean, it's just the science is just advancing so rapidly on how we can examine these tissues and learn about what's going on in these tissues. So really exciting to hear about this whole new field.
Thank you so much, Veronica. Veronica, we really believe that if we are right, we could make a difference. There is also a chance, guys, that we are missing so much of it. We are trying to build these models from really huge amount of data. There's so much that needs to be done. It's almost like we try our best to like focus on a few things and like have papers one at a time so that others can build on our work as well. But I do think that there's a lot of space. For example, I just showed you guys there's so much changes that occurs, right? But I need to do it. What changes that occurs? We want to do this precise high resolution thing, but we are not there yet, in my opinion. So these are almost like early signs of thing, in my opinion. Some may work, some may not work. Yeah.
And again, I think this is going to be recorded, so you may hear from people afterwards. I heard from several people who said they were going to watch the video later on because they couldn't attend because of SRI. Shuo, anything else?
Yeah, all clear. It's very great.
It's fantastic. I mean, I think it's really, this is where multidisciplinary idea exchange is so important. You know, there's people doing one thing in the cancer world, and it's so applicable to the oncofertility world and evaluating tissues and how they're behaving. And you might not even see or hear from each other unless somebody reaches out. So thank you so much for sharing your work.
Of course, Veronica. Veronica, on that note, I do want to ask, we want to get access to the
Mary Solomon, who's here, she's our ERTA. They're busy extracting clinical data because we want to give it to you with some clinical data. But did you need the clinical data or just the images and age?
No, we do need the clinical
Yeah,
data.
so they're extracting all kinds of clinical data. I think we should have it for you in a couple of weeks.
Okay, sounds
the
good.
files.
We are looking forward.
Perfect. And then for anybody else who's here, again, if you reach out to me, we have a committee that reviews the proposal. You just put together a one-page proposal, and then we can give you access to the images. We have so far had 2 groups, Sanju's group, and then Jasmine and Melbourne. They're also reaching out and using this data for a very different project. But I think this is really exciting. This is what the oncofertility was all about to begin with.
Thank you so much, Veronica, for putting this together. I do appreciate it.
Great. Thank you, everyone.
Thank you. Thank you,
Thank
Veronica.
you. Thank you again.